Hello children. Today we are going to start with a new topic, that is development of a pollen sac or a microsporangia. Yesterday we have discussed that if the anther is bithecous, then it is called to be tetrasporangiate, and if it is monothecous, then it is called as bisporangiate. That means two microsporangiums will develop. Now, how these microsporangiums develop from the very first beginning cell, and which is generally called as the anther primordium. I have always told you that if something initiates from a single cell, that cell is always called to be a primordium. For example, leaf originates from the leaf primordium. The flower originates from the flower primordium. In the same way, this what we call microsporangium develops from the anther primordium. To understand this, if we have this anther lobe which is bithecous and having the 4 microsporangiums, in the young stage it must be a single cell like this. Single cell. And immediately a one layer of what we call cells, which are generally the hypodermal cell, that means just below the epidermis, behave as a archisporial initials. So these becomes archisporial initials. These are archisporial initials. Archi words come from primitive. The first one, sporo means sporogenous tissues. That means the spores, that is the microspores, are getting to develop from these initials only. Now, if many layers or many initials that participate to form a microspore, then this type of development is called as eusporangiate. The term eusporangiate is used. But if only a single cell develops into the whole microsporangium, then it is called as leptosporangiate. Most of the angiosperms show this type of development, that is eusporangiate. Most angiosperms does this thing, that means whole of the archisporial initials participate in formation of the microsporangium. Now, what happens? Anther primordium is there. It is at a very young stage. Then it gets mature. A strip of archisporial initials are there. They undergo divisions. To understand that, what type of what we call divisions takes place, we should know the two kinds of divisions. One is called as periclinal division, another is called as anticlinal divisions. In periclinal divisions, if the cells are like this is one layer of cell, it cuts into half like this. That means this becomes a outer layer and this forms a inner layer, right? But in case of anticlinal division, if this is the layer of the cells, the divisions takes place by this means. That means in this, the number of cells in a row increases. This is called as anticlinal divisions, and periclinal means the number of the cells remains the same, but we get two layers, the outer layer and the inner layer. These both divisions are simple mitotic divisions. So a strip of archisporial initials very firstly undergo the periclinal divisions. That means we are getting a outer layer and a inner layer. So what do we get is outer layer and the inner layer. The outer layer behave as a primary parietal layer. As it is a result of mitosis, it will remain diploid. And then it undergoes periclinal and anticlinal divisions. That means when these layers, undergoes the primary, peri and anticlinal divisions, they form many layers in a rows. So what do we get? In the previous lecture, you remember that the anther layers are there, and these are called as microsporangial walls. What we have done yesterday was epidermis was formed, endothecium, the inner layer, then innermost, that is the middle layers, then inside the tapetum is formed. As these are undergoing only the peri and anticlinal division, that means only and only mitosis is going on. Therefore, epidermis will be diploid, endothecium will be diploid, middle layers will be diploid, but in case of tapetum, we have discussed that it is polyploid. This means karyokinesis is happening, but it is not followed by cytokinesis every time. So we get a cell which is a polyploid. That means it can be 3N, 4N, 5N and so on. We have discussed the functions of the tapetum yesterday. So these layers are formed from the outer, which is outer layer, from the archisporial initials by undergoing the periclinal divisions, and this outer layer will be called as primary parietal layer. Parietal itself means, that is from the periphery the word has been derived, that means the outer side. Now inner layer, this inner layer, that means again from the archisporial initials when the periclinal divisions occurred, we had got the two layers, outer and inner. The inner one is called as primary sporogenous layer. But of course, only mitosis is occurring here till now. That means it is diploid. In the same way, again the mitosis will occur to increase the number of cells. This becomes sporogenous tissue inside the this tapetum layer. It is getting inside because the outer layers have formed these things. The inner layer will form this now. This sporogenous tissue again undergoes mitosis just to increase the number of cells, and these cells will be called as now the pollen mother cell, which is called as microspore mother cell. Microspore mother cells means this cell will give rise to a microspore, generally called as, in a short form, MMC. We know it as a meiocyte, that it will undergo a meiosis to form the male gametes. So this is again a diploid structure because till now no meiosis has been occurred. So this is again 2N. This was also 2N, diploid. Now in the very initial stages, the pollen mother cells, they remain attached with these connections. These connections are the protoplasmic connections. These are protoplasmic connections. And soon they form a callus outside. They get somewhat separated now, and these behave as the pollen mother cells. This, when they form a callus ring, callus outside, that is beta glucan, that forms, that will undergo meiosis. After meiosis, we know each cell will give rise to 4 cells. Meiosis is a reductional division. If this is a diploid structure, when it undergoes meiosis, it forms 4 haploid structures. When it undergoes meiosis, it forms a tetrad. This tetrad is called as microspore tetrad. These microspore from one cell are formed 4 only, but they still remain attached to each other by a callose formation. This microspore tetrad, you must be seeing here that 3 are visible, but 1 is at the back side. So tetra means 4. Tetrad we have got. After what you call some time, the dehydration takes place, the callose dissolve. Here I want to tell you that this callose, what you call deposition, is again what you call degraded by a callase enzyme. This callase enzyme is again produced by tapetum. One of the function, that means it produces callase enzyme to dissolve this callose deposition, which is surrounded by a tetrad, microspore tetrad. Immediately when the callose dissolves, they form a single microspore, gets detached, and we get a microspore, that is N. This all is happening inside this, that is the anther only. They remain inside. The microspores are formed inside. Microspore undergoes a precocious germination. That means in situ germination. That means the germination initiates inside the anther only. And immediately it forms a pollen grain structure. These details of the precocious germination and how the pollen grain forms the male gametophyte, we will discuss in the next section. But till now what we have done is, from the primary sporogenesis, first mitosis forms sporogenous tissue. Second, mitosis undergoes to form a pollen mother cells to increase the number. Then they undergo all the pollen mother cells, they undergo meiosis to form a 4 tetrad like structure, which is called as a microspore tetrad. Callose dissolves and they form microspores. Microspores undergoes the precocious germination, again a very important thing which is asked many times in exams. Precocious germination, that means the in situ germination, that means the microspore starts germinating inside the anther only and it forms a pollen grain. Here I want to tell you one more thing, which is many times asked in your entrance exams. That means if a tetrahedral like this is called as tetrad. If tetrahedral structure, that means 3 in front and 1 is back, that is called as tetrahedral tetrad, microspore tetrad, and it is found in dicots. It is a feature of a dicot. If isobilateral structure, that means 4 when you see in front, that is seen in monocots. Sometimes this type of tetrad is also seen, that is T-shaped, 2 above, 2 down, linear, 4 in a row, and then decussate. This is somewhat different from this. This structure is different. One is at the back, but still the structure is different. That is why the name decussate is given. So these are some types of tetrads. One thing here to remember is there is one species called Aristolochia. It is a one plant species which forms all these types of tetrads. All these types of tetrads. This you have to remember. This is the species which forms all these types of tetrads. So, in the next section, we will discuss how the microspore changes into a pollen grain and then to the male gametophyte. That's all.